Okay, thank you. I'm just going to go ahead and share screen, and hopefully this will be seamless. Okay, do you see a full screen of my PowerPoint?
Yes.
Great. Okay, and I'm going to minimize you all. Okay. Good morning, everyone. So thanks for having me. I really appreciate it. The paper that we're going to be discussing today was part of my PhD research at Oregon State University under the advisement of Dr. Stephen Good. My project was entitled Streamwater Microbial Communities as Hydrologic Observation, Insights Gained from an Investigation of a Novel Data Set. So this research project was inspired by an observation that we and others have made that hydrologic research seems to be a bit stalled on a number of fundamental understandings. Despite decades of research by some of the world's greatest minds, there's still much we don't know about the movement of water through catchments and specifically in heterogeneous landscapes. So it's clear that new approaches are needed to advance the field in a really meaningful way. Another observation that we made was that DNA sequencing seemed to be everywhere. And it's true that DNA analysis has become much more accessible across research domains as the cost is just a fraction of what it was just 20 years ago. We also both understood a couple of key things thanks to our strong high school level knowledge of biology, that microbes are astoundingly diverse and DNA
I'm not sure if you can hear us, Dawn, but we lost your sound.
Yes, I'm here, and her video is frozen.
Maybe we'll try to turn off our videos.
Yeah, let's do that.
I'm going to send her a text suggesting she log in again.
Thank you, Hoshi.
Maybe you could send her an email because I don't know if she's getting the message.
Yes. Okay.
Hi, Yuan. We just lost our speaker, so we're waiting for the speaker to come back online.
Oh, okay.
I think we should sell this break for advertisement and make
Yeah.
some money.
All right. Do you want to advertise our workshop? We're making progress, by the way, guys. We more or less have the draft flyer out. I mean, we haven't sent it out, but it's been designed. The first circular, and the advisory board has sent some weighed in with some comments. And we're at the point where we're just trying to create a short list of invited speakers and start reaching out to them. But probably within a few weeks, we'll be sending out the first circular so people can start signing up.
Hi, everyone.
Hi, Dawn. Sorry we lost you.
I'm really sorry, but it seems like my internet connection just failed there for a second.
Sure.
Thanks for letting me know. I would have given that whole talk and not realized that I had lost you. Okay,
No
so
problem.
let's.
I do have a question when you bring that slide back up.
Yes. As soon as I figure out how to bring that slide back up. For some reason, I don't see my, oh, there we go. There we go. Is this where I left off, or where did you lose me?
When
Previous
you were
slide or
showing
here?
the DNA sequences, the cost.
Oh, yes. How do I do this? There we go. There,
Yeah,
this one?
Yeah, so you might start here again. But the
Okay.
question I have is, so you have this interesting linear relationship up till about 2007, and then this drop off, which presumably was some kind of technological breakthrough. And then it becomes linear again. And I'm just curious if you can speculate what the limitations are right now on.
Oh, that's a good question. Yeah, I don't know what the limitations are right now. As far as like watching the cost continue to fall, is that kind of what you mean? Like
Mhm.
why it's holding steady?
Yeah.
Yeah, I don't know. I can't answer that question. And in fact, this is a slide borrowed from my advisor, Steve. And so I'm not even sure if this represents like the actual like cost, including lab cost of like extraction, or if this is just the cost of like sending it out for sequencing, like how much you would be charged for such a thing. So I can't say.
That's okay. Just curious.
Sorry. I will look into it though. I'd be happy to
Yeah.
get that answer in email. Okay, so then, so you wanted me to start at this slide, or did I get kind of through
We got
this
this.
part?
Yeah.
Okay. So we launched into some really naive and rudimentary machine learning, and using a published data set of microbial sequences from a set of 6 large Arctic rivers. We found that we could actually train machine learning algorithms on the summer sequence data to predict monthly discharge and return interval throughout the entire year for those 6 rivers. So the predictions weren't perfect. So what you're looking at here in these plots is the average relative error between our predictions and the actual observations. They weren't perfect, but they were better than just using sort of this benchmark mean of the other 5 rivers. So it seems that the genetic data does add some information. So we hypothesized from there that there was some connection between hydrology and microbial communities that we could capitalize on to advance hydrologic research. And so we dug into the literature and we found that microbial ecologists were already starting to identify relationships between microbial freshwater communities and the
Sorry,
terrestrial
Dawn.
environment. Yes. Did you lose me
Sorry.
again?
No.
Oh, okay.
Could you just say what kind of machine learning algorithm you were using?
Yeah, so we used SVR for that WRR paper.
Support vector machines?
Correct.
Okay, thank you.
Yep. So we learned from the literature that freshwater microbial communities are actually seeded from microbes that are passively dispersed from the upslope environment. We also found that community structure was statistically related to characteristics of the catchment. So in this paper by Salvio et al., they reported that the hydrologic characteristics like river mile, so the distance downstream, and the size of the catchment explained more variation in community structure than physiochemical parameters like pH and dissolved oxygen, and comparing communities sampled along the entire continuum of the River Danube. So thus we began a project to employ the stream microbiomes to learn about hydrologic function and to shed new light on some of the unresolved questions in hydrology. So we started collecting our own data in the summer of 2017. We collected 61 samples near active stream gauges across the Willamette and Deschutes basins. So this Google map, yes?
Don, just a short
Yes.
question.
Yes.
Is this also related to some work I heard of where people use artificial DNA as, tracers with unique IDs? Did you try that as an alternative, or is the biome just better because it's a tracer in situ already?
Exactly. So we believe that because it's naturally occurring, that it is actually sort of like a fingerprint of the processes that are happening in the catchment. So This is kind of getting into like this study that we're finishing up now, but we're sort of thinking about it as a hydrologic tracer. But because it's a biological tracer, it is showing, so it responds and interacts with the environment in a different way than just geochemical tracers, because it sort of has these biological interactions. And we believe the benefit of using this naturally occurring DNA, first of all, is that we don't have to like do any preparation. So we just go out and collect samples. And also that we believe that because it's naturally occurring, it is sort of integrating all of this information from the catchment in this like really high dimensional data set. I haven't looked at a lot of papers about the DNA that you're talking about. So you're talking about like not necessarily eDNA, but like actually like artificial
Artificial
sort of
DNA, yeah.
That's like sort of released as like a tracer.
Yeah. It apparently proved to be a little unstable, so it was then hard to capture it in the DNA sequencing later on.
Yeah, so that's also a thing that we learned, is that DNA is, it's not very stable. And so we had a goal of processing all of these samples within 20 minutes of collecting them. So that's sort of like the magic number that we have is, so we can sort of like stabilize it in the field using just a chemical buffer that we add that sort of like freezes the community as is. So that when we take it actually into the lab and extract and sequence it, we are actually sequencing like the community that we sampled in that moment.
Mm-hmm.
Yeah, good question. Thank you.
Mm-hmm.
So the first initial samples that we collected are along the Willamette and Deschutes basins here. So you can see the Willamette in the west side and the Deschutes on the east side. So they're separated by the Cascade Mountains. Oops. I'm using my roller and getting ahead of myself. So they're similar in area, but drastically different in annual precipitation and discharge at the outlets on the Columbia River. So precipitation is about 3 times higher in the Willamette, and discharge at the Columbia is about 6 times higher in the Willamette than in the Deschutes. So we collected our samples over 2 major multi-day campaigns in late July and early August of 2017. And I like to joke that we pioneered a very specific technical sampling protocol called throw a bucket over the side of the bridge and pull up some water, which is pictured sort of here. And then we standardized collection as much as we could by just attempting to pull from the center of the stream and without disturbing the riverbed and the sediment. So we basically just have like free floating bacterioplankton samples. So we collect the water in the bucket and then we pump it through a peristaltic pump to avoid contamination, and that's pictured there on the left. It's collected in these 0.2 micron filters, and then we store the filters at -80 degrees until processing. Then we extracted and amplified the 16S rRNA genes, which is how we isolate the microbial constituents from, say, eukaryotes like plants and animal DNA. And then we send them out for Illumina MiSeq sequencing. We then prepare an abundance table of each of the amplified sequence variants, or ASVs, which I will show you here. So then this is basically the product that we have, and this is what we feed into our analysis, is just this abundance table. So we have a series of sites along the top here, and then all the unique microbial taxa on the sort of the columns. And then it's just this huge matrix of the abundance of each of these and each of the sites. So we rarefy the data set to a constant number of sequences per sample to standardize the sampling depth across sites. And this rarefied number of sequences is sort of different for each analysis, depending on sort of like how many samples we have and which samples we're using and how much data we can afford to lose, because oftentimes we'll lose some sites that don't meet the rarefication threshold. So in our first analysis, we found that microbial community assemblages were shaped by catchment scale geomorphology and climate in headwater catchments. So there's this box plot here that shows the relationship between differences in catchment characteristics and differences in communities based on the type or the classification of the catchment characteristics. And so here you see in these small headwater stream, we have these really strong correlations between geomorphic, climatic, and to some degree land cover characteristics and differences in communities. So since smaller catchments are typically headwater streams and upper reaches, this difference in correlation between small and large catchments may be evidence of a largely terrestrial derived microbial community that's shaped by the catchment that transitions to a stable freshwater community in lower reaches. So we published those findings in Frontiers in Water in 2020, which is how we came to the second hypothesis, which is the paper that we're discussing today. So the hypothesis that we wanted to test for the current paper is that the streamwater microbial community carries quantifiable hydrologic information. We reasoned that if microbial community is shaped by these catchment characteristics that also shape hydrologic regime, then the streamwater microbial community would therefore encode hydrologic information about the catchment. So to test this hypothesis, we had two main tasks. So to quantify the relationships between microbial taxa and hydrologic characteristics at different scales, and then to compare patterns in the relationships between the microbial taxa and the hydrologic characteristics.
Dawn, a short question.
Yes, yes.
I know that Laurent Pfister in Luxembourg, he used diatoms to analyze sources of water, and it seemed like a lot of work to manually classify, the different diatoms under the microscope. So your approach seems to be much more doable on large samples, but can you compare the two methods?
I can't. I'm not familiar with that study. I've seen it, but I'm not familiar with the details of it.
Okay.
So yeah, I can't really compare it. So he looked at them, or they looked at them under a microscope and identified them like by their sort of taxonomic relationships.
By their skeleton structure, basically.
Okay, so it looks like they were maybe looking at like, I guess like phenotypical like physiological characteristics, and then relating that somehow. Like, was it like a statistical relationship, or was it more functional relationships that they were looking at? Do you know?
So they looked at the, you can't actually call it skeleton, I guess, but at the microcalcium structure of these diatoms to see where they were from. So they were collecting this in source and target areas and try to make sense then of where the water came from. And so it looked interesting, but also like a lot of manual labor. So that's why I thought your ways of detecting using the DNA sequencing might be much more promising in that sense.
Yeah, I mean, so the great thing about this, about the DNA, is that first of all, for every sample, you get thousands of sequences, like you get thousands of hits. And so there's just so much information per sample, which allows us to do a lot of like statistical analysis. And so the paper that, for this paper here, this study here, we were looking basically at just like the statistics. That's how we kind of started with like the machine learning, is we weren't really looking at like the functional or ecological role of any of these ASVs. We were just looking at them as sort of like a series of 0 and 1s, like it was just like a high dimensional data set. But then on the follow-up study, which we're writing up right now, we do actually start to look at sort of like the functional roles and the taxonomy of these individual species. And we are looking at whether we can make some inferences about sources of stream water during a precipitation event. So it seems like we're kind of starting to get into that phase, but it's really difficult because so little is known about these microbial taxa that it's, like I think out of like the thousands of taxa that we identified, there were only, there's a database where you basically can just like feed all of your sequences into this database, and it returns the taxonomy. So in that way, yeah, there's very little like manual labor because it's all sort of already done, and we're capitalizing on the work that's already been done. But the microbial community is so diverse that so little is known about these individual taxa that I think there's like a lot more of that will be possible in the coming decades as we sort of like build on our existing knowledge and start to look into these individual taxa to some degree. But, you know, right now there's the knowledge that we have about these taxa is basically limited to sort of like larger classifications and is pretty broad. But I think there is definitely potential for that moving forward.
Mm-hmm.
I can talk about that at the end. I have a couple slides I can show if there's time. So for this MI mutual information analysis that we did here, we added samples from an additional basin, the John Day Basin, which is off to the east here. And those samples were collected in the summer of 2018. So the annual precipitation is similar as the Deschutes, but discharge is much lower. So about 1/5 of the discharge that we have in the Deschutes. So the inset here, so the inset here shows the number of unique and shared microbial sequences detected in each of the basins. So for this analysis, just to reduce the analytical complexity, we focused on the 256 taxa that are here in the middle of this Venn diagram here. So the 256 taxa that were detected in all three samples in at least, or in all three basins in at least one sample. So we wanted to examine and compare relationships between the stream microbiome and hydrology at different time and flow scales. So we analyzed 3 groups of metrics. So the daily discharge, which is the observed discharge at time legs of 2, 4, 6, all the way up to 30 days before sampling. We felt reflected the short-term environmental conditions and perturbations. Mean monthly discharge, which is just the observed long-term mean discharge across an approximately 10-year period of record, we felt represented more long-term means and characteristics of catchment function. And then finally, we looked at seasonal flow durations, which is just the 5 and 95% exceedance probabilities in each season and annually to sort of capture the extreme flows in the catchments. And consistent with can't find the word I want, nomenclature elsewhere, we have large Q representing the absolute observed flow and then small q, the flow normalized by the area. So in all, we looked at 76 hydrologic metrics. So to quantify the relationships between microbial communities and the metrics, these hydrologic metrics, we used entropy-based metrics. So in the version of Shannon's entropy, we used log base 2 so that our resulting quantities are in units of bits. So here we analyze entropy of random variables X and Y, where X is the abundance of some ASV across all sites, and Y is the value of some hydrologic metric like mean January discharge across all sites. So we develop histograms of observed mean flows across all sites and then use those probabilities to determine the entropy of that metric. So what we looked at, of course, is the mutual information, which is the area where these two areas of entropy overlap. So you're familiar with this, but I'll just go over it. So the mutual information, of course, is the reduction in uncertainty in one random variable that comes from observing another. So in our analysis, we wanted to determine the reduction in uncertainty of a flow metric that comes from observing the abundance of some microbial taxa. Because we wanted to be able to compare the amount of mutual information across flow metrics, we needed to normalize the raw mutual information scores because they represent the reduction in various values of entropy for each of the different metrics. So we normalized the mutual information by the entropy of the flow metric, which is in the box here, and results in units of bits per bit, which is also the percent reduction in uncertainty. So here is what we found. So we have a heat map of the significant values of normalized mutual information for each ASV and each hydrologic metric. And ASVs are presented in order of decreasing abundance, with the most abundant on the left, ASV1, and the least abundant on the right. The top panel shows absolute discharge and the bottom is discharge per unit area. And then we have daily discharge metrics in the top row, monthly in the center, and seasonal high and low flows in the bottom. White areas indicate no significant information. And for this study, we determined significance as mutual information scores that were higher than the 90th percentile of 1,000 random shuffles of the abundance of the taxa for each flow metric. So some interesting things to note from this plot. First is that every hydrologic metric, so every row, there is some information somewhere, which means that at least one ASV in the microbial community encodes information about all of the metrics that we analyzed. Another interesting pattern is that most of the information is concentrated in the more abundant taxa, so on the left-hand side of the plot. So in fact, in the plot on the left, we see that there is a significant linear relationship between the log of abundance of a taxa and the amount of information it shares with hydrologic metrics in all three categories. So across all discharge metrics, there's an average of 15.6 ASVs that share significant information with a given hydrologic metric. And for absolute discharge, the median value of shared information was 0.163 bits per bit. And for discharge normalized by area, the value was 0.150 bits per bit. So because we observed strong relationships between microbial community and absolute discharge, we focused the remainder of the analysis on absolute discharge metrics only.
So Dawn, a short question. All the
Yeah.
binning schemes were then kept the same for all variates, target and source, I guess.
Correct. Yeah, we used Scott's rule of thumb, and I think we looked at like each metric and each ASV, and we found that like between 5 and 9 bins was appropriate for all of the histograms. And so we ended up just using 5 bins across all metrics and across all ASVs.
And you were continuously univariate, so one predictor versus one target, or did you also try them in teams?
No, we didn't. And that's a good question. No, so for this study, it was just one ASV, one hydrologic metric. So we sort of like, yeah, we just did one at a time. As far as like looking at pairs, I'm not really like, so I think that would be like sort of like conditional mutual information, right? Or like conditional, we looked at that, but it was just like so much data. I didn't know how to tackle it. And because we looked at, we didn't really look at like the functional roles of these ASVs, I wouldn't have really known how to group them, except to maybe group them by larger like phylogenetic groups. So instead of looking at species, we would look at something like class or order, and then maybe we could group them that way. But we're also finding in the research, and this is something that we're kind of looking at in our current study, is that these taxonomic relationships are, you know, they're based on evolutionary history, and they're not necessarily indicative of like functional similarity. And so we were afraid that some information would be lost if we had too coarse of a sort of grouping scheme. So we started here with just like individual taxa and individual metrics. And I kind of thought like the next piece, the piece that I couldn't do with the data set that I had, that I've been talking to Allison a lot about, is that I would really like to be able to look at the total amount of unique information across the entire community. So what is the value of the community for each hydrologic metric? So basically doing like a information decomposition. But as of right now, we don't really have like a method to do that with this amount of variables, I guess. I think Allison has proposed a method that I just like haven't had the time to like look into, but I think that that would be an interesting route.
May I just ask one more question? Can you
Please, yeah.
give me one example of, say, a functional relationship, or like why should the abundance of one of these organisms be related to a hydrological state? Can you give me an explanation for one, just so I get an idea?
So a functional relationship. So for example, some of these taxa we've identified as being really related to like nitrogen fixation, which might indicate that they have some sort of like symbiotic relationship with plants, which might indicate that they're like terrestrial taxa. And so maybe this suggests that there are like surface derived taxa. Another one might be
Yeah, we lost them again. I'm gonna email her.
This is very interesting. I mean, it makes a lot of sense that biology. Ah, you're back. Perfect. No sound.
no sound.
Oh, now you're back. Yep.
Okay.
We hear you now.
Okay. Sorry about that. I'm not really sure what's going on. My internet connection looks okay.
It happens to the best of us. It's happened to
Okay.
me. It's happened to everybody here. So don't worry about it.
Okay.
You were saying.
So I was saying about did you hear the bit about the groundwater?
No.
No, okay. So I was saying, so some functional relationships that might be of note would be something like if we identify taxa that are, that's not what I want. Sorry, I want to bring up there we go. If we identify taxa that are related to nitrogen fixation, so that might suggest that there is some relationship with like a terrestrial community, and maybe this indicates that there is some surface contribution or subsurface contribution. Or if we have taxa that are known to be oligotrophic, so they thrive in low nutrient environments, maybe this would suggest that there is like some groundwater contribution or something like that. So the idea is that sort of, I think for the most part, the idea is that we can sort of source these taxa based on inferences about their sort of relationship with the environment. Does that answer your question? Okay.
Yes, thanks.
Okay. Okay, so then next we looked across the metrics within categories and compared the amount of information shared with different time legs, seasons, or high and low values. So we basically broke these down, each of these rows, to look at metrics within categories. So for each of these categories, we compared two metrics of shared information. So the median value of shared information across the entire community, and also the number of informative ASVs for each metric. So here we have the box plots illustrating the median value and distribution of normalized mutual information across time legs, where the green triangles represent the number of informative ASVs and corresponds to the y-axis on the right-hand side. So for daily discharge, we find that the median value of mutual information over time fits a second order function with a peak in median information around 16 days prior to sampling, which is illustrated by the red line down here. And we find that the number of ASVs sharing significant information also fits a second order function with a peak in the number of informative ASVs at around 22 days. So basically, when we sample, when we collected our summer samples, they seem to be most informative of the discharge about 2 to 3 weeks prior. And then for long-term mean monthly discharge, we find that the highest median value of normalized mutual information was in February. though there was no significant trend in time. So we had this peak here, but there's no trend through the months. The number of informative ASVs did follow a second order function with the maximum number of ASVs in winter, which is interesting given that we collected our samples in June and July. So it's reasonable to suppose that this greater amount of shared information with winter flows is more related to the magnitude of the flow rather than a strictly temporal trend. So we experience a Mediterranean climate here where we have wet winters and dry summers, and winter corresponds to characteristically much higher flows and more variable. So thus what we're seeing here might reflect a relationship between the amount of shared information and flow volume. However, we observe no pattern in the amount of shared information with high versus low flow durations, which is what I'm going to show here. So we have the seasonal high flows in dark gray and low flows in light gray, and the number of informative taxa in dark and light blue. We observe no significant difference between high versus low flows as far as the amount of mutual information or the number of informative taxa. So from this study, we concluded that the streamwater microbial taxa do encode information about stream discharge at different temporal and flow scales, and that most information is contained within a small number of the most abundant microbial taxa, which is actually great news because those are the easiest and the cheapest sort of to sequence. And that summer microbial taxa share the most information with long-term mean winter flows.
Question, Dawn.
Yeah.
So you're seeing a strong correlation with wetness in your catchment. I'm curious whether you explored correlation with energy available also.
Oh, interesting. With energy available, like net ecosystem productivity or something like that, or
I'm not sure how you quantify it, but I'm just generally thinking that, you know, when we do aridity index, we're looking at a trade-off between energy limited and water limited, right? So water would be potentially be a necessary resource, but the complementary resource would be energy available for the biota to do their functioning. And I'm just curious if there might be another dimension that's not perhaps completely encoded in your data sets yet.
So your idea, so I think that might be different than our objective. So the idea here would be to like learn, your idea is to sort of learn about like microbial community structure and like assembly.
No, I'm saying that you're seeing increased activity or reduced activity or whatever, and then you're seeing correlations between that activity, unless I'm misunderstanding you.
Yeah, I don't know. I wouldn't say that it's necessarily related to activity.
Okay.
So it's like the abundance of certain taxa. It might not necessarily be like greater abundance. It's just that the abundance, like the probability of some certain value of abundance is related to that hydrologic metric.
I see.
Does that, so not necessarily that they're always like increasing. It's just like the amount of mutual information, the median amount of mutual information for each taxa is greatest in February, but not necessarily that the abundance is greater.
Okay.
Does that?
Yeah, because
does that
when I
clarify?
think of abundance, yeah, no, maybe I have to think about it some more. When I think of abundance as being, you know, related to both resources and energy available to reproduce, and so I was maybe confused there.
Yeah, so it's, and also, so like when we talk about abundance, it's, I should say, I'm a little loose with the term. What we really mean is relative abundance. So like basically it's like its proportion of the community, because each of these samples are rarefied to the same number of taxa. So they each had, I think for this, I think it was 1,450 for the sample that you read about, because the way that you sort of sample, you know, like the way that we sample is like we our very standardized sampling protocol was to pump water until the filter clogged, and depending upon local conditions, that volume of water is different across sites. However, there's been, you know, in earlier papers we published that there actually is no strong relationship between like microbial diversity and the volume of water.
But is there not, doesn't relative abundance indicate a competitive advantage, or am I again misreading that? Oops.
She's gone again. Frozen.
Yeah.
My video
There you go.
there.
Yeah.
Are you with me now?
Yeah.
Yes.
Okay. I don't know if I can screen share if that's still like having video. Maybe I'll just chat and bring up slides as necessary. So that's a great question. And that's sort of like at the heart of a lot of this research right now is, you know, there's sort of like two major factors shaping microbial communities in these freshwater stream systems, right? So there's dispersal effects or mass effects, which is just like the microbial being carried into the stream and like passively added to the stream through, you know, like basically hydrologic conditions. And then there's also like species sorting, which are these like sort of competitive effects and like the environment sort of selecting for particular taxa. And so in headwater streams, we find that it seems that mass effects or dispersal effects seem to be overwhelming species sorting effects in the upper reaches, just because of the nature of microbial ecologists refer to this idea of like residence time, which is a little bit different than hydrologists think of it, but it's basically like how long a microbe or a community has been in the stream. So how long a parcel of water has been actually in the water channel. And at some point, those sort of like dispersal or mass effects become overwhelmed by this sort of species sorting, but that happens sort of like in lower reaches, like as the community develops. And so I think what we're seeing here, and that's why I think we're seeing such strong relationships between the microbial community and hydrology, is because I don't think that we're necessarily seeing, I think we're seeing less species sorting and more dispersal, which I think sort of favors hydrologic information. That's my read on it. I don't know if that's like, you know, so I think that's why we see this like strong relationship between, you know, hydrology and catchments, is because these communities are shaped largely by dispersal from the terrestrial environment. And in lower reaches, that relationship starts to weaken as sort of species sorting and competitive and predation and all those things start to sort of take over and shape the communities.
That's helpful. Thanks.
Then I have a question.
Yeah.
So in those factors of viability of the species, do solar energy or insolation favor some of these species so that the abundance could be related with a signature in the annual cycle or daily cycle of these in this species?
Yeah, so that's a great question. So yes, there are seasonal cycles in microbial communities. So it seems like, and they're probably related to all sorts of things. That is like probably in the microbial literature, but one of our co-authors, Byron Crump, has done a lot of work on looking at these like sort of seasonal communities, and they find that there's like these core seasonal communities, like there's a core summer community and a core winter community, and this has been shown in like freshwater systems all over. And that there's like these sort of like spring and fall are kind of these like transitional communities that sort of transition between the two. And this is shown like mostly statistically through like PCOA and like cluster analysis. I don't know that they've gotten into, at least in the literature, you know, like in the research that we're looking at, there's not a lot of looking at like specific taxa. It's sort of like looking at communities as a whole and sort of like their general structure. And so that's another reason that we feel like the microbial community could be really good to study, is because that there is some stability in communities. Like it's not, there's like a good balance, I guess, of like stochasticity and like stability in these core communities. So they are shaped, they're not random, I guess is what I'm trying to say. They're not like these random amalgamations. They're there is some structure to them and how they assemble, that is shaped by some factors that we believe are hydrologic and environmental factors.
I see. So that means that the microbial community prevails over a species. So you're sure that that community it's gonna be seen in the tracer, right? In the samples that you take.
Like if we were to go back and sample again next year, are you
Yeah.
or you're like across basins?
Mm-hmm.
Yeah, so I don't know how much of, so there's one study, actually, I'm gonna see if I have it available on my slide before I try and. I think I saved it. No, I didn't. I just I zapped it. Shoot. Yeah, so there was a study done by URycki, was actually one of the motivating studies for our paper, and actually where we got the data set for that machine learning paper, where he looked at those 6 Arctic rivers, and he found that there was a lot of consistency in the taxa, even among those rivers that were, you know, like geographically separated, but hydrologically similar. And that there was like some synchronicity in the seasonal cycles among those rivers. So I think there's I mean, I guess we're kind of hopeful from this research that there would be like some taxa that we could consider like flags of some sort, so that they would be indicative of some hydrologic condition, or I don't know, some sort of like process or dynamic in a system that we would be able to like apply in other catchments outside of our study area. But I think we're kind of a ways from that, but that's like sort of the greater objective, is that that yeah, we would be able to identify some taxa with some consistency in some study areas that would tell us something about other study areas. If that's kind of, I sort of interpreted that as being sort of like where you were going with that question.
Yeah, thank you, Dawn. This is very interesting.
Yeah, you're welcome.
So I have a question that I probably should have wrapped my head around by now already. So you have the mutual information between these flow metrics and abundances, and you're finding that like the larger abundances are the ones that tend to be the more informative of all those flow metrics, like so that like heat map that you showed. And you're normalizing the mutual information by the entropy of the flow metric, which makes sense. So then you get this like reduction or fraction of reduction in uncertainty. So the reason that the higher abundances tend to have the higher percentage reduction in uncertainty is that also partly because the higher abundances tend to have higher entropies themselves, so they have more information to give? It's like if you normalized by those instead, it would have made a little bit less sense in terms of like the fraction of reduced uncertainty, but would you see that the ones that had the highest entropies also had the most information? They gave the most information because they have the most information to give.
Yeah, I think so. I think I understand your question. And I think that one, I think I do have a plot for. I think I'm just going to share, see if I can get away with sharing this quickly. Yeah, that's a good question. So I think, and so when we talk about like the highest abundance, we're talking about like the highest abundance across all, not necessarily like on a daily, like on a per sample basis, but across all sites. And I think higher abundance is related to it also just being captured in more sites. So it just being more common in general. So it just shows up in more sites, and therefore, yes, I think it has more information to give. I hadn't thought about it in terms of it being like the entropy of that abundance, but I think that's probably it. Like we didn't really know how to describe that, but other than saying like, yeah, if it appears more places, there's just like more, there's just like more dynamics in the abundance, and it's like more relationships between the hydrologic metrics if it shows up in more samples. And that's kind of what this plot was designed to sort of show. So the reviewers were curious if we had lost information when we sort of cut our data set down to 256 taxa from like the 4,000 that we identified. And they're like, well, couldn't you be losing a lot of information? And we're like, well, we don't really think so, because we think in order to have mutual information, you have to have like a lot of relationships, like it has to have shown up in a lot of sites. And so I did this quick box plot to look at. So I looked at the mutual information among all the taxa and all the hydrologic metrics, and just added up the number of relationships. And so what I found was microbes that showed up in all 3 basins. So this x-axis is the number of basins in which I identified a taxa. So this is the 256 here that showed up in all 3. So the number of relationships between those taxa was just higher than for microbes that showed up in only 2 basins or in 1 basin. So I think this is just like sort of another way of coming at the sort of like what you are getting at, that like the entropy of these taxa is related to the amount of information they carry. So your idea is sort of that, like, if I normalized by the entropy of the taxa rather than by the entropy of the hydrologic metric, I would learn that, like, what would be my conclusion from that?
Yeah, maybe that's not a good idea, but maybe like on that x-axis, you know, you rank it from the abundance, like the most abundant to the like 100th most abundant. If you change that to be the entropy instead of the abundance, you know what I mean? So it's like,
yeah,
maybe the number 1 most abundant taxa also has the highest entropy. Sorry, I sound really terrible. This is going on YouTube too. So maybe you'd see the same pattern, like maybe the highest abundances correspond to the ones, well, because like you said, you have just fewer zeros in that matrix because they show up more often in all of the watersheds, like all of your samples. So it might end up just being the exact same result.
oh yeah, but I like that way of thinking about it much more. That's a really good idea. Thank you. Like a light bulb finally like clicked on about that heat map. That's a really cool way of thinking about it. Yeah. And that would also be a way of maybe, that could be also a way of like a more informed way of sort of trimming down our input is to choose the microbial taxa with the highest entropy rather than just like the most abundant. Other questions, ideas?
Were you done, sorry, with your presentation?
Yes. Yeah, that was it.
Okay. So what do you see as the next steps?
Oh, great. I'm glad you asked. So for the next, I'm going to try sharing screens one more time here. So the next thing is we are looking at, so instead of looking across basins, we looked at one stream here. So we collected samples across a major precipitation event at the beginning of the wet season here in Oregon. And so we're looking at sort of the relationship between microbial community structure in time as it relates to discharge, as a way of, we're hoping to sort of gain insight about the sources of stream water to during like event flow conditions, and like sort of like the contributions and contributing area, and sort of wish I had a more broad way of characterizing that. Yeah, so just like looking at sort of individual taxa and finding relationships between phylogenetic groups and discharge, and seeing if we can see some actual relationships between how the microbial community responds to hydrologic conditions at this like really high temporal resolution. So we collected daily samples for about 2 weeks. And we found some really interesting stuff. So writing that up, and hopefully it'll be out soon.
Do you foresee, do you have any ideas about how you might use this information to integrate it into coupled hydrological, biological models?
Yeah, so I think the key is going to be in these sort of functional relationship, or like the sort of like the functional ecology of these microbial taxa, and looking at sort of identifying how they relate to the environment. I think we also did for this, I don't know if I have the, yeah, I guess I don't have it. We did like a concentration discharge analysis with the microbes over this precipitation event, and we found that there were actually some microbial taxa that had significantly positive or negative relationships with discharge over the course of this event. And so we feel like that shows that there are actual characteristic responses of larger taxonomic groups. So like gamma proteobacteria seems to be diluted with discharge. And so we think that that has something to say about like whether these are sort of like freshwater associated, you know, just like any, I guess, geochemical tracer, like that sort of like hints at, you know, the sources of water to a stream, and like how those dynamics change over the course of an event. And because we're able to find relationships in more broader communities, like proteobacteria is a large phylogenetic group with like, you know, thousands of taxa. And so those would be relationships that we would definitely be able to see in other catchments, and maybe be able to make some inferences there, this sort of like, again, like indicator species. As far as incorporating them into models, I feel like that's like really far off. Like, I don't know how they fit into models just yet. I think right now it feels like it's very much like sort of qualitative
Conceptualizations.
Yeah, conceptualizations. Yeah, and just sort of like, you know, sort of probing what kind of information is available using different sort of analytical techniques. You know, we did like the machine learning, and we did some sort of classical, you know, concentration discharge. And so we're just kind of tossing all kinds of analytical techniques to see sort of like where we can get the most information from the communities.
Unless I missed something, you're primarily focused on surface waters at the moment, is that right?
Right. So that, yeah, for this, it was, yeah, surface water samples, streams and rivers, just those 3 basins.
Any thoughts about extending this to groundwater samples?
Yeah, we just don't have that data right now. Like that would have to be like manually collected. I think what we'd like, I think the broader goal would be for it to be more widely applicable. And so I think trying to find ways to capitalize on public data sets would be really, to me, that's sort of like the next step, is like NEON, you know, so NEON is collecting a bunch of this kind of data, and they're doing some soil water samples as well as freshwater, I think. So I think that is a way to do it. I guess I think of myself as like an engineer first. And so the idea of like making tools rather than just like sort of the academic, you know, I guess I'm really interested in like how it applies more so than, thanks everyone for those that have to take off. I lost my train of thought. Yeah, so I guess we're interested in like sort of building a tool that's like more widely applicable, and groundwater sample, like the surface water samples that we collected are relatively easy, logistically speaking. But it was like, you know, a week in the lab to extract,
Sure.
you know, 25 samples, and then, you know, cost quite a bit of money to send them out. So, and then groundwater samples, like it would be easier to collect them from existing wells, I think. I used to have this dream that I was going to like collect all of these samples in these watersheds so that I could do sort of this like variable contributing area analysis and like do this sort of like tracer with this like sort of end member analysis. But it's just like a tremendous amount of field work and
Sure.
and analysis. Yeah.
I mean, it just strikes me, of course, it's early days. It strikes me, you know, we start by just measuring magnitude, quantities of water.
Mhm.
And starting to measure chemical constituents. But it would seem like a more complete analysis, and you would hope that in the future you would have some well-established set of indicators, and you would measure also chemical and biological constituents so that you could build a comprehensive understanding of the system. But probably it's a little far away. And we need the kind of work you're doing right now to help understand what are the most informative indicators, or maybe it's not that complicated. Maybe you just collect what you collect, but you still have to analyze it and look for something. So you have to know what you're looking for.
Yeah. Yeah. And it seems like there's a lot of, that's the thing, is like the field is just developing so rapidly. You know, it's like NEON is collecting this kind of data, which is really exciting, but also it seems like we're already moving on to the next thing. So it seems like they're moving on from 16S amplicon, which is sort of like this like taxonomy, like where you sort of identify the species of all these different sequences. And now they're moving into metagenomics, which is really exciting, where you look at, instead of grouping sequences based on their like evolutionary relationships, now you group them based on like identification of functional genes. So for example, like this is the gene that's responsible for nitrogen fixation. So even though these two species are evolutionarily not related, they both fix nitrogen. And so by looking at that and grouping them that way, that there would be like a whole, that information might be much more valuable. And that is like is a pretty new analysis, and it's a little bit different and a little bit more expensive. So, but I think like that's a thing that I think like in 10 years, like we'll be having those conversations instead of 16S. It's sort of like old news now.
That's great. Thanks.
Then I have one more question. So
Yeah.
Is it fair to say that when you sample DNA, is it more successful when you use it in pristine water rather than in polluted water, just because some toxins are able to create a decay in the community? Or how do you deal with those anthropogenic disturbances?
Oh, that's a great question. Some colleagues, so the short answer is no, I don't think it matters, because, you know, microbes are so diverse, they can live in any sort of community. And so I think as far as like a successful sample is one that just like gives us a lot of information. Like we're not looking for necessarily particular taxa. And some of our colleagues, did they look at I was gonna say they looked at microbial groups, but maybe they didn't. I think we were going to. They did some like sort of machine learning on, I guess, I want to say it was microbial groups, but I don't think they were. Anyways, the idea is that we collected like chemical samples and microbial samples at all these like various levels of disturbed freshwaters right here locally. And they were able to, I think this is not relevant now that I'm saying it. I think this is like not what I want to say. Forget that study, never mind. But the idea is that that microbial communities are so diverse, and actually like, you know, some of these microbes are known for like breaking down these like really toxic anthropogenic chemicals in the environment. And so I think that these like sort of environments and conditions are just selecting particular taxa, to me just adds to their informational capacity. And so I don't think that I can't imagine a scenario in which you would like collect a dead sample, like where you just wouldn't find like that it was so toxic there was no microbial community. Like I think there just always is. And so I think it's actually like really valuable as potentially an indicator of things like that, and like, you know, the influence of some particular toxin or you know, whatever it is, sort of the breadth of it and its persistence and all that, I think could be something that you could gain from the microbial community.
Yeah, it's awesome to see how persistent are microbial communities and that you are able to trace them. Yeah, even if they are in very difficult conditions. Thank you, then.
Leela, what's our next meeting going to be? Or do we have somebody lined up yet?
No, so we don't have a presenter for the next session. So yeah, this is an open invitation to everyone who wants to, yeah, share their recent work with us.
All right, we'll look into that.
Yes.
Dawn, I'm going to approach our seminar coordinator to try to get you to give that talk to our department. That would be very interesting.
Oh, thank you. I appreciate that.
That's for sure. So, I don't know if you know Yang Song. She's also working on sort of this interface between hydrology and biology and ecology. Yeah.
Oh, cool. Yeah, no, I'm not familiar. That would be great.
She's actually the coordinator, so I think she'll be very excited. So,
Well, that'd be cool. Yeah, I would love to talk. That would be great.
all right, good. And I don't know if Leela has informed you about this, but we are organizing another workshop next September in Karlsruhe. It's actually in Germany, at the Zugspitze, on the highest mountain in Germany, on bridging information theory with computational science and domain science. So are you on our mailing list?
Yes, I am. I think I saw something about that. Yeah.
Okay.
Yeah.
So stay
That
tuned.
sounds pretty cool. Okay.
Because yeah, if you have the time, energy, and information to attend, this could be a nice addition also to the discussion.
Awesome. Thank you.
Very good. So Leela, 3 weeks from now.
Yeah, see you then in 3
All right.
weeks. Okay,
Okay. Thanks again, everyone, for your attention and your
Thanks, Dawn.
questions.
thank you, Don.
Take care.
Bye-bye.